So the partner study is a clinical trial using Remicade in conjunction with standard therapy for hepatitis C. Leading up to the partner trial, there's a phase of about 5 years of looking at things that I'm going to get you through it, because I think any trial is probably irrelevant if you don't know what led to the trial. We already heard that biologics and viral hepatitis is a topic of interest, and the number of biologics have grown, and there has been reports of hepatitis B reactivation. There has been reports of even patients who went into liver failures as they were placed on biologics in the setting of hepatitis B. Part of the reason, if you look at this picture taken from a paper about 5 years ago in New England Journal of Medicine, talking about all different cytokines involved in health and illness in liver disease, and many of them, whether it's TNF or IL-6 or TGF-beta, all involved in the process of liver inflammation or liver regeneration. When anti-TNF came to light, there were some. This is one a trial in which looking at ALT in patients who received Remicade early in the clinical trials, whether they had RA, Crohn's, UC, ankylosing spondylitis, and psoriasis, and there were not a lot of signals as to toxicity issues in terms of just elevation in ALT in reaction. So these patients received placebo. This is Remicade, so ALT 1 to 3 times upper normal limits. The more severe toxicity, as we define, is greater than 3 times and certainly greater than 5 times. So if you look at here, it's very uncommon to have, at least based on these old studies, to have significant ALT elevation in response to Remicade therapy. And when chronic viral hepatitis is a unique and different, it's probably different than fatty liver disease. That's more recently is becoming more and more of interest in everyone who uses biologic, and we heard already about both of them. This is a slide you saw. The hepatitis C is where I'm going to be talking about, and it is, for us as hepatologists, it is a disease in which leads to advanced liver disease. This is a liver cirrhosis, and this is hepatocellular carcinoma. So, but when I came into this issue of anti-TNF in hepatitis C, we started by few reports that suggested elevation in TNF level in patients with HCV infection, it seems that these elevation. This is one study that we performed in which it basically compared to, we looked in whether in the serum or in the tissue you will have elevated TNF levels in patients with hepatitis C compared to normal liver tissue, which is in the middle. But there were additional reports that suggested that elevation in TNF level in the serum or in the tissue may actually be associated with resistance to the therapy of hepatitis C. So the higher the TNF level in patients' blood, the more likely they will fail antiviral therapy with hepatitis C. So maybe there were some suggestions as to TNF maybe blocking the potential beneficial effect of antiviral therapy of interferon. And in fact, that may have made sense when you look at hepatitis C viral kinetic when you treat the patient. So when a patient is placed on interferon, the hepatitis C RNA goes into 2 phases of decline before eradication of the virus. And the first one is very rapid decline that is being directly related to the antiviral effect of interferon. But the more important phase in which actually is necessary for viral eradication is the phase 2, which is a slow decline, happens over weeks or months and lead up to eradication of the virus. It is believed to be T-cell mediated elimination of infected hepatocytes with the virus. Most studies showed that about probably 30 to 40% of hepatocytes are infected with the virus at the time of chronic hep C infection. This is a very old paper from the rheumatology literature published in JCI, in which in this paper they demonstrated that actually TNF-alpha may block the ability of T cells to activate. And so so these are T cell looking at as the TNF-alpha is increased, these cells are T cells gradually exposed to greater and greater amounts of TNF-alpha. The T cell stimulation index seems to suggest that these cells become sluggish, don't respond to antigens very well, a response that actually can be reinstated by giving the patient anti-TNF-alpha or giving these cells anti-TNF-alpha. So all that together, we actually put a hypothesis at the time, and this is, I'm talking about the year 2000, so about 10 or 12 years ago, in which we said, okay, so if some of these information are true, then our hypothesis was that a liver infected with hepatocytes will, these HCV will have its antigen that are recognized by antigen-presenting cells associated with Th1 activation, production of interferon gamma or other T cell cytokines that is associated with elimination of infected hepatocytes. Along the way, interferon gamma may activate macrophages associated with production of TNF-alpha locally in the liver or systemically that may actually inhibit now T cells. So it makes that what's supposed to be a successful viral elimination cycle, it becomes somewhat blocked or unsuccessful elimination cycle. So very simple, very naive hypothesis, but we decided to test it because we had at the time one clinical trial in IBD patients, 60 of them that underwent therapy with etanercept for inflammatory bowel disease at Mayo Clinic. And I identified 13 of those patients in that trial in which had abnormal ALT at entry, of which 3 or 4 actually had hepatitis C infection, and 8 out of the 12 actually normalized ALT while they were on anti-TNF, and we saw no a change in HCV RNA when they were on anti-TNF, on etanercept. So we said, okay, so we have some data to suggest that it's reasonable, it's no safety issues, at least based on this particular trial, and we have all this data that may suggest some benefit. Additional issues related to TNF and clearance of hepatitis C is the insulin resistance that have been highly associated with lack of clearance of hepatitis C while on treatment. And TNF-alpha is a major mediator of insulin resistance. So we said so either by its effect on T cells or by its effect on insulin resistance, both of which may actually be associated with lack of response to interferon therapy, and therefore maybe we will see some benefit. And this is just one experimental mice model published in 2004 in which they showed that the core protein in hepatitis C actually induces hepatic insulin resistance and high TNF-alpha that you can actually fix or reinstate into a normal insulin sensitivity using TNF antibody. And in this paper they used the Remicade to do that, infliximab. So with all that in mind, we designed our first trial, which was actually a phase 2 clinical trial that we published in 2005, in which we randomized, initially we started 50 patients, but some dropped out before receiving any drug. So the final outcome that 19 received etanercept plus interferon and ribavirin, and 25 received placebo plus interferon and ribavirin. And it was a randomized trial, double-blind. And what we found, simply I'm cutting through a lot of issues in the trial, but essentially that actually what we anticipated might happen, it did happen in this trial, in which we see that if you look at, this is the group that received etanercept plus standard therapy, this is the group that received only standard therapy. So whether you look at HCV RNA negativity, normalization of ALT, or patients who had both endpoints met, meaning clearance of the virus and normalization of ALT, whichever endpoint you look at, the patients who received the etanercept plus standard therapy did better. And in terms of toxicity in that trial we found no liver toxicities in this trial, no increase in frequency of adverse effect between the etanercept versus the placebo group. We saw the typical side effects associated with interferon and ribavirin. And in fact, this was the most intriguing to us, that some of the side effects were less, particularly GI side effects, flu-like symptoms were less frequent in patients who received anti-TNF, and they were similar. Actually, there were data from multiple sclerosis and others, models of diseases in which interferon is used, that have been suggested a link between the side effects of interferon and TNF-alpha. So we said, okay, this is really unanticipated, but we were happy to see it. So this is all leading up to the partner trial that has been completed, have not been published yet. And this was a follow-up, more a phase 3 following the phase 2 trial, in which the aims of it were to investigate whether adding infliximab, in this case, to standard treatment of PEG interferon alpha 2b and ribavirin improved SVR, SVR meaning viral clearance or viral cure, decreases the rate of fibrosis in the liver, or improve safety profile. And it was a randomized, blinded, controlled, multi-center efficacy and safety study. Total subjects were 150 recruited from the origin on 10 sites, so we anticipated about 15 patients per site. Interim analysis after 50% of subjects completed 6 months of study. We have a DSMB chaired by Lenny Calabrese at the time. These were the sites selected for the trial. So we had 2 California sites in both LA and San Diego, Dallas, Houston, Florida, Pennsylvania, anyway, and then 3 sites here in Cleveland, Ohio, 3 different hospitals in Cleveland, Ohio. This was the design. So we took all patients with one genotype. Hepatitis C come in different genotypes, so we took one genotype to control for that. So half the patient received standards of care plus placebo, half received standard of care plus infliximab. There was an interim analysis, as I told you, mostly as it related to safety, and everybody received what we consider to be standard therapy for 48 weeks. And the primary endpoint was complete viral eradication, or what we call it in hepatology, SVR, sustained virologic response. At the time when we did when we were designing the study, we actually looked at some of the data on FDA post-marketing hepatotoxicity of TNF, actually with the help of Lenny, who was able to obtain the data. So July 8, 2003, these were the total number of cases in which supposedly there may have been liver toxicity. The additional search suggested that severe liver injury is really not that as common as what looking at all possible liver injuries, so 110 cases, 41 and 7. And when we actually looked at liver cases and we tried to actually exclude those what we believe to be autoimmune liver disease reactivation and so on, the numbers are even smaller. So considering the number of patients at that time point that had been exposed to anti-TNF, we felt that the likelihood of any significant liver injury would be low, so we proceeded with the trial. This is after the rigorous exclusion. Actually, most of the cases were reported. We felt that they were mainly related to underlying autoimmune hepatitis that was reactivated. And this is what the trial, so we screened 220 patients to eventually enrolled, instead of 150, it was 149 patients. So for 149, I think there were 4 patients who never received drug, dropped out before they received any drug. So we have 73 in placebo group and 73 in the infliximab group. The results are simple, and I'm almost done with my presentation. We found really no difference in the rate of sustained virologic response between the arm in which the patient received a standard therapy and placebo to that of patient who received standard therapy plus infliximab. And these rates were very much exactly what we expected if someone received standard therapy based on published data. There were also no differences in the spectrum of adverse event or SAEs between the two arms of the studies. So what have we learned? And this is my final slide. I am not sure what we have learned at this point. So we have, yeah, I decided to say that only in the last slide, so you don't tell me not to give my presentation. What we learned is, one is, at least so far based on 2 phase 2 and phase 3 trials, we did not identify any specific safety signal in this population when you combine anti-TNF, whether etanercept in the first trial or infliximab in the second trial, when it comes to using this medication in the setting of Hep C. What we also, I don't know if we learned, we are now puzzled with these discrepant results between the 2 trials. So is it a difference between phase 2 and phase 3 clinical trial? Actually, I took some time to look at how often data from phase 2 trial is positive that turned out to be completely negative in phase 3. And I was actually, to my surprise, very, very high. So apparently, if you look at oncology, there are studies on that. If you look at oncology trials, 60 to 70% of oncology trials that showed efficacy in phase 2 will show no efficacy in phase 3 clinical trials. And if you look at all other specialties, whichever you pick, is anywhere between 40 and 60%. So it is very common in which phase 2 trial would suggest something, and this is one of the biggest challenges to pharmaceutical industries, is when to act on a phase 2 trial and proceed to a phase 3 trial. And there are a number of reasons for that. We can discuss as much as you want. The second issue is, we know from other diseases that there are differences between TNF antibody and between etanercept. So infliximab and etanercept are not identical. There are other disease models in which there are differences in efficacy and outcome between the 2 products. So we really, at this point, I'm not sure if it is a agent-specific finding that we had in phase 2, or it is phase 2 versus phase 3 clinical trials. And I will stop here.